In this video, we'll take a look at mutation. Mutations are permanent genetic changes to DNA. Errors sometimes occur during self-replication of the DNA molecule. When these errors occur in the sex-determining cells, they are passed on to offspring. Rarely, mutation results in a favorable change that makes subsequent generations better equipped to deal with their environment. Evolution occurs. More frequently, mutation results in unfavorable changes that make the offspring profoundly unsuited to its environment, or the offspring may not develop at all. There are two types of mutation, chromosomal and gene mutations. We'll take a look at chromosomal mutations first. DNA makes up chromosomes. Two chromosomes code for the same set of characteristics. This is called a homologous pair. Take a look at the human karyotype to help you remember this. Aberrations of this pattern result in mutations such as translocation, deletion, duplication, and inversion. In deletion, a piece of a chromosome is absent. It's actually just been lost. This makes the chromosome significantly smaller, and there's a theory that says that the Y chromosome is simply an X chromosome that's actually lost a significant segment of genes from it. In duplication, more than one copy of a gene is present. So if this was the original chromosome, you can see that on the new chromosome, there's actually an extra copy of the original gene. This is responsible for some forms of Down syndrome. During inversion, part of a chromosome breaks loose and rejoins with the ends reversed. So if we label these genes A and B, during inversion, they're actually in reverse position, and so are all the genes in between. During insertion, a portion of one chromosome gets inserted into the sequence of another chromosome. And during translocation, chromosome pieces are exchanged between two non-homologous chromosomes. So this is taking it one step further than insertion, where the chromosome pieces are actually exchanged between two non-homologous chromosomes. Now, it is important to note that exchange between homologous pairs is actually normal, and it's a process called crossover, which actually results in more genetic diversity. It's desirable. This is not. Let's take a look at a few gene mutations. In gene mutations, an actual nucleotide may be deleted. So if the normal sequence of bases is TAC, GGA, ATG, a deletion would result in a different pattern. For example, if the T was removed, we would end up with the sequence ACG, GAA, TG. You'll notice that all of the bases shift over by one spot. And what this results in is some very different triplet codes, and in fact the third one isn't a triplet code at all, so it would not code for any amino acid. The result of this is pretty serious. We end up with a non-functional polypeptide. In an addition mutation, an extra nucleotide is added, and it also results in a non-functional polypeptide, and you can see that it has pretty devastating effects. If an A was added at the beginning of this sequence, it would frame shift all of them over by one, and we'd end up with three completely different triplet codes and a leftover base at the end that coded for nothing. Just in this small example, we would get three completely different amino acids. In substitution, one nucleotide is substituted for another. So if you look carefully at the normal sequence and the substitution sequence, you'll see that they're not all that different. The only difference is that this G has been replaced by a C. Sometimes this results in a functional protein because, as we know from the table of mRNA triplet codons, some amino acids are actually coded for by more than one triplet code, and if this letter replacement actually results in the code for the very same amino acid, it might not actually have any effect at all on the finished protein. However, it may actually code for one different amino acid. So while this amino acid, and all the rest of the amino acids would be the same, the one coded for by this sequence, if it is different, can result in devastating effects. Only one amino acid chain results in sickle cell anemia, and in this case valine substitutes for glutamic acid, resulting in abnormal hemoglobin that results in very serious life-altering consequences for the person who has the genetic mutation. Just to give you sort of an idea of how a single deletion can result in a frame shift mutation, let's take something that we're more familiar with, just a sentence that actually makes some sense. The big dog bit Ted and ran off. If one letter is deleted, this would be like deleting one base in the sequence, it becomes nonsense information. The bid og it edda neder anof means nothing. And in the very same way, a deleted base from a sequence of DNA results in a polypeptide that means nothing to the cell and is non-functional. Try these for yourself. If the sequence of DNA is TAC, GGG, CTC, TAG, CGA, GAT, ATT, the corresponding mRNA sequence would be AUG, CCC, etc. Use your table of mRNA codons to try to figure out what the amino acids are that are coded for by this sequence of mRNA. Well, AUG is the start codon, and it always codes for methionine. CCC codes for proline, and the rest are shown as well. Now, these we got when we looked up the mRNA codons, not the DNA triplet codes in the table. Pause the video and try the other two examples shown here, and then check back and see if you got the matching sequence. Here are the sequences of amino acids that you should have come up with. Now, what causes these changes to chromosomes and genes? Well, DNA changes are caused by environmental mutagens, and there are two types of mutagens. They can be chemicals or they can be radiation. Chemical mutagens include some food additives, hallucinogens, pesticides, and many of the chemicals found in tobacco. Some chemical mutagens result in a DNA adduct, which is a piece of DNA covalently bonded to the cancer-causing chemical rather than another complementary base. This process could be the start of a cancerous cell or carcinogenesis. Not every mutation leads to cancer, but when they do, we call those mutagens carcinogens. The second environmental factor that can cause damage to DNA is radiation. Non-visible short wavelengths such as X-ray, gamma rays, and those emitted by radioactive elements penetrate cells and cause mutations to DNA. Ultraviolet radiations with wavelengths above 260 nanometers are absorbed strongly by bases, producing pyrimidine or thymine dimers, which can cause error in replication if left uncorrected. So, for example, if adjacent thymine molecules are bonding to each other instead of to their complement, what happens when protein synthesis occurs?